Good morning, everyone. My name is Caroline. I'm a master's student at PPGC-Bio, and I will present my project titled Study of the Cytogenotoxicity of PBS Nanoparticles in 3D Human Keratinocyte Spheroids. First, let's talk about nanoparticles. They are very small materials with high surface area, high reactivity, and a size similar to biological structures. Because of these characteristics, they are used in many fields. Among these nanomaterials, we focus on PBS nanoparticles. PBS is a biodegradable and biocompatible polymer with good thermal and mechanical properties. PBS can also be modified with molecules like glutamic acid and itaconic acid to improve its behavior, including degradation and cell adhesion. But, like all nanomaterials, their small size increases reactivity. This can lead to toxic effects, so it is important to study their safety. Previous studies showed good compatibility of PBS in 2D cultures, but 2D models do not represent real tissues very well. For this reason, 3D models are better. 3D spheroids reproduce biochemical and biophysical signals, allow better cell-cell and cell matrix interactions, and maintain more stable cell phenotypes. Keratinocytes are excellent models for toxicity studies. They are sensitive to environmental changes and widely used in research on skin biology, healing, toxicology, and tissue engineering, especially in 3D systems. So, no reports were identified in the literature investigating the interaction of PBS nanoparticles in 3D models of human keratinocyte spheroids, particularly regarding the evolution of their cytotoxic, genotoxic, and molecular effects. So the hypothesis is that PBS nanoparticles are cytocompatible, do not induce cytogenotoxic effects, and do not alter the expression of genes related to the viability, proliferation, and differentiation of keratinocyte spheroids. So, the general objective of this project is to evaluate the cytocompatibility of PBS nanoparticles in 3D keratinocyte spheroids, focus on cytogenotoxicity and gene expression. For the experiments, the nanoparticles will be produced by Professor Ariane Group, and the spheroids will be formed using ultra-low attachment plates. We will also characterize the spheroids by measuring diameter and cell viability with MTT. We will perform physical-chemical characterization of PBS nanoparticles with dynamic light scattering, Raman and infrared spectroscopy, and scanning electron microscopy. Cytocompatibility tests will include cell morphology, flow cytometry, and mitochondrial activity. Genotoxicity will be evaluated using the COMET assay, and gene expression will be assessed by RT-qPCR, examining genes related to proliferation, differentiation, and stress response. During the 4 months of the project, I am also defining a protocol for reliable spheroids formation. I am using ultra-low plate and plate with agarose. As expected results, we hope that the 3D keratinocyte spheroids will maintain viability, morphology, and DNA integrity after exposure to PBS nanoparticles, showing good cytocompatibility. These results will help identify safe concentration ranges and support the use of PBS nanoparticles in future biomedical applications. Thank you.